Hello and welcome to Toto's Collection Anatomy session. So here we're doing an introduction to histology so that we get the basics. So what is histology itself? This is just a study of tissues, and if we look at man, there are 4 major tissues that he must have. So this means that any other tissue that we might come across should fall under one of these 4. Now, who loves semen? Who loves to see men? So these are the 4 major tissues that you have. So you have connective tissue, you have muscle tissue, you have epithelial tissue, and then you have nervous tissue. So these are the 4 major tissues. So any other tissue that you might come across should be able to fall under one of these guys here. So before we start studying these tissues, it is not a quick thing such that you open up a person's body and then you get a sample and then you take it directly to the microscope. That's not how it works. So sometimes special procedures are required in order for us to study our tissue in depth. Though there are some cases where we can just get the tissue as it is and then put it under a microscope. For example, if you were to get blood. Blood is just gotten as it is, probably with some little of staining, and then put on the slide, and then it is studied for microscopy. It could be as simple as that, but most of the times it's not as direct as this. There needs to be those preparatory stages that a tissue will have to go through in order for it to be viewed properly under a microscope. So quick question, blood. I've given an example of blood. What type of tissue is it under these 4 tissues? Semen. So here we have the histological techniques. So these are the procedures that are taken in order for us to have our sample prepared. So this is the first part here. We're going to get our sample from our tissue. Cut it into a manageable size, quite a small piece of it. And then the first step that we're going to do under our histological technique is called fixation. Now here, this is where we fixate our sample. What does it mean to fixate your sample? This simply means to denature all the proteins that are in this sample. You're going to denature all the proteins that are in his sample, because usually what happens is this: you cut off a tissue from its support system, or from its supply of nutrients and oxygen. It's supposed to start dying, and after it starts dying, its entire structure is supposed to start withering. After that, it's just supposed to start degrading. One of the things that aid in the degrading of the tissue are the enzymes that the tissue actually has. So there could be enzymes inside the tissue that will start eating up the tissue itself. So to prevent all these degradation, all these denaturing, all these breakdown processes that would happen to a tissue after you cut it off from the main organ, you are supposed to fixate it. You're supposed to undergo the process of fixation. So what example of a fixative can you think of that is used under this fixation process? That's a homework for you. Next up, we do have this thing called dehydration. So just from the term itself, dehydration, you already know what that means. We're removing water, but here we're not removing water at once. It's not a quick thing such that boom, water is removed. Water is removed steadily and gently, and what we do use to remove water from this tissue or this sample is basically alcohol. So we have these 3 jars. So we have jar 1, jar 2, jar 3. So each of these jars contain your alcohol, but they do contain alcohol in different percentages. So you add your sample to the first jar that contains alcohol, but this alcohol is of little percent, somewhere about 50%, and then you later take it to a sample that has a slightly higher concentration, so let's say somewhere about 70%, until finally you can add your sample to a jar that has a concentration of 100%. That's how you dehydrate your sample. So you do not jump directly to the alcohol concentration of 100. No, you start slowly. You start 50, you go 70, and finally you reach to 100% concentration of alcohol, and that is how you dehydrate your sample or your tissue. That's the next step. After dehydration, your next step is clearing. What is the purpose of clearing? In clearing, we just want to make our sample transparent. Okay, so what are we going to use? We are going to use a chemical called xylene. This is an organic compound called xylene. It is going to be used in the clearing process to make your tissue transparent. Okay, after you've made your tissue transparent, you are going to put it in hot, or I can say warm, or I can say boiling paraffin. Here you put it in paraffin. This is in the infiltration process. So here you are allowing your sample to be infiltrated with paraffin. Okay, so at this temperature of 52 to 60 degrees Celsius, and then after you've infiltrated your tissue with paraffin, you allow it to cool down, and it's usually cooled down in these solid blocks, and that process is overly known as embedding. So you have infiltration, where you have that paraffin that's liquid and warm or hot, and then after that you cool that paraffin down together with the sample inside the paraffin, and then you put it in these blocks, and that is called embedding. And then after you've embedded it, now you get the whole cube. You get this whole cube and put it in a machine called a microtome. This is the microtome. You put the microtome there. As we can see here, we do have what they're calling a paraffin block, so meaning the entire paraffin block that you previously had, let me use a different color, the entire paraffin block is going to stay right about here. Now this microtome is used to cut the tissue into minimal sizes, so it's going to cut the tissue together with the paraffin in this way. So imagine this is your block, something like that, and then we do have our tissue somewhere about there. So what is going to happen is that when you put this block inside your microtome is just going to slice this part here, part here, and then you're going to have something like a very thin sheet of your tissue together with your sample like that. So this is going to be like the paraffin, and your tissue is going to be there, and then you're going to put this on a slide, and then you're going to add some staining, and then you're going to view it under a microscope. That's basically the histological technique. That's the histological preparatory process, and that is how you get your tissue ready for examination under a microscope. Not so complicated, right? So those are just the steps that you need to know, beloved. You need to know these. A quick run through before we go to the next part of this histological process thing. So remember, first part is we cut it from the organ itself into that small size. We fixate it. What is the purpose of fixation? Halting all biological process, denaturing all proteins, stopping all enzymes from degrading the tissue. Next up, we dehydrate it. We remove the water. What do we use? We use alcohol. How do we do it? We do it in slowly increasing concentrations of alcohol from 50, 70 into 100. Next up, we clear our tissue, make it what? Transparent, using what? Xylene. After we made it transparent, we infiltrate it with what? Paraffin. How do we do that? We heat up the paraffin, put the tissue inside the paraffin, and that is the infiltration process. Lastly, we embed it, simply meaning we cool down that paraffin, and we have our block solid paraffin with the tissue inside it. Next up, we put it into this microtome and cut it into our desired size, and that is basically the whole process. So next up, we are supposed to look at how we stain these guys.